Thank you, Tim. Hello and welcome, everybody. My name is Berenike Dietrich, and I did my thesis on the recruitment ecology of the stalked barnacle Pollicipes. In the following, I'm going to give you a quick overview about the background of the study, the approaches that we used, what were the outcomes, and what we can conclude from them. The focus of this study is the stalked barnacle Pollicipes, which is a sessile crustacean species. It inhabits the intertidal zone of exposed rocky shores along the northeast Atlantic coasts, and they are suspension feeders with a slow growth rate. The growth rate is also known to vary strongly between individuals, which is important later on. In Spain and Portugal, or at least in some regions, they are also harvested as a highly prized seafood delicacy. Their habitat, the rocky intertidal zone, is characterized by constantly fluctuating conditions such as the tide and different disturbances. Disturbances, in this case, are to be understood as wave action, storms and other natural phenomena, but also the impact of humans, for example, by scraping off the seafood, leaving cleared patches on the rocks. As for most sessile species, a good place to settle is most crucial for survival and therefore strongly contested. These disturbances create new opportunities for recolonization and thereby also enhance the biodiversity of this habitat. These recolonization processes are also referred to as ecological succession. Here, certain key species compete with each other and eventually replace one another in a stepwise manner. And the settlement of larvae also belongs to the processes of ecological succession. The cyprid larvae, which is the larval stage responsible for the settlement in barnacle species, has been observed to respond to ecological cues regarding to their place of settlement. Most preferably, they settle or they attach on the adult stalks, which in the light of sustainable fisheries poses a problem, as it leads to high numbers of juveniles as bycatch when they grow on top of the adults, which are then harvested for consumption. In this study, we wanted to explore the ecological factors other than the conspecifics that might be important for the choice of settlement for the cyprid larvae. To approach this objective, we did a photo analysis, and we used photos that were taken in the frame of the European Biodiversa project. The photos were taken in 3 different locations along the Asturian coast, which is in northern Spain. In each of these locations, 12 metal quadrats with an inside length of 35 centimeters were attached to the rock and photographed every 3 months over a time span of 2 years, from July 2017 to July 2019. This results in a total of 36 of these metal squares with 9 pictures of each square. What we then did was we used the georeferencing function in QGIS to level these 9 pictures, for example, to neutralize the angle in which the photographs were taken to make them better comparable. In the next step, we went through the pictures chronologically to identify when new recruits of stalked barnacles had settled solitarily, meaning that they were not associated with the already existing stalks. We then marked them with a 2 by 2 centimeter square, as you can see here in green. Additionally, we used the random point function in QGIS to create controls of spaces where no new recruits had settled. For each of these quadrats, we went then 3 months back in time, meaning to the picture that was taken previously to the one that they were detected in, to analyze the ecological conditions in which, at the point of time in which we assumed that the larval settlement had been taken place. The analysis consisted mostly of a semi-quantitative species cover estimation of these 2 by 2 centimeter squares. We did that on 6 levels. So either a species was absent, barely present or roughly 25, 50, 75, or 100% abundant. In this example, you can see that species were found in different abundances in the little square. Additionally, we also recorded the season and categorized the surface of the underlying rock according to its roughness. This was then transferred to a spreadsheet, which was the basis of the preliminary statistical analysis that was conducted in the study. Because of the growth rate variation of the stalked barnacle juveniles, we did not only record the ecological conditions 3 months before detection, but also 6 months, 9 months, and 12 and 15 months before the detection. And for each of these chronological intervals, we then developed a statistical model in R. In total, we were able to identify 228 solitarily settled recruits with 14 different taxa living in their proximity. Most of them, we were able to identify them to a species level, and they consisted mostly of different algal species, limpets, mussels, and acorn barnacles. The most explicit effect that we could find on the recruitment frequency of the stalked barnacles was the surface structure of the underlying rock. As you can see here, we have a category of smooth rock, a category of small fissures, and a cracked surface. And we can see that with the higher roughness of the surface, the recruitment frequency increases. This was actually consistent in all our models, which leads us to believe that our models can predict the data more or less accurately. Other significant effects were discovered for these 3 species, and we believe that this is also due to structural properties, but in this case of a natural, of a biological nature. For example, these 2 red algae species, Corralina elongata and Lithophyllum tortuosum, are both calcareous algae that form small erect shapes above the rock. And for both algae, the recruitment frequency of the stalked barnacles increased with the algal abundance, but it decreased at 100% abundance cover, or at 100% coverage, which leads us to believe that the larvae prefer to settle on the margin of these algal patches instead of in the middle of them. Then we have the Mediterranean mussel, Mytilus galloprovincialis, which is also harvested for human consumption in this area. And it also has a significant effect when crossed with the factor season. We know that mussel abundance itself is strongly dependent on season, but we found that small patches of mussels 3 and 6 months before the detection of a new recruit seem to be beneficial for the recruitment rate. Whereas big mussel patches seem to be beneficial 12 and 15 months before the detection. Corresponding with the life cycle of mussels is to assume that the mussels that were there 3 to 6 months before detection have been alive at the point of larval settlement, whereas it is very likely that the mussels, or the big mussel patches, 12 to 15 months before the detection of a recruit, have been already dead at that point of time. This might lead to the assumption that a combination of little competition of either dead mussels or only small mussels, and the structural properties of the either alive mussels or the leftovers of the dead mussels, for example, shell pieces or byssal fibers, are a good combination for the recruitment. Other ecological cues for the settlement might also be the coloration of the substrate. Earlier studies have found that stalked barnacle larvae or other barnacle larvae prefer to settle on darker surfaces much rather than on lighter colored surfaces. In our study, the red algae, or the abundance of the red algae, Rhodotamiella floridula, increased the settlement of the recruitment of the stalked barnacles, even though it doesn't have any structural properties as it is a filamentous algae. It has a dark red color. On the other hand, we found that the acorn barnacles, which build cement structures but have a very light color, showed little effect on the settlement. Here it is very essential to note that acorn barnacles are associated with later successional stages as well as a higher trophic level, and therefore might pose a competitive threat to the stalked barnacle larvae. And this could be also the reason why they didn't prefer to settle on the acorn barnacles. Further, we had assumptions about the effect of height, not only on the recruitment frequency of stalked barnacle larvae, but also on the growth rate of the juveniles. Unfortunately, we could not record the height of our samples because of weather conditions. But interestingly, we found that Nemalion helminthoides, another macroalgae that is very common in the upper intertidal zone, meaning higher above the sea level on the rocks, is associated with the models of the ecological conditions 12 to 15 months before. This might lead to the assumption, or this might support the assumption, that the juveniles in the upper intertidal zone grow much slower and can therefore be detected only later. Now, to put these outcomes into perspective, it is important to remark that this study served to develop hypotheses much rather than to confirm them. And the key aspects that can be taken out of this study and should be further examined are that the solitary settlement of stalked barnacles is higher in or on abiotic or biotic structures on the substrate surface. And maybe in future research, can show that the increased availability of these surfaces can land on the adult stalks. This information could then help to improve the sustainability of the management, which in this region is managed or controlled by the local fisheries, the cofradías, and the local government, the Dirección General de Pesca Marítima del Principado de Asturias. And further consequences could also be the regulation of the mussel harvest, which in this region is much less strictly regulated than the stalked barnacle fishery, which might change when it gets more evident that the mussel abundance impacts the recruitment ecology of the stalked barnacle. Now, I would like to give thanks to my supervisor, Professor José Acuña, and to Katia Geiger and all of the staff from the Departamento de Biología de Organismos y Sistemas in Oviedo, to the Corona Barnacles crew, to my friends and family, of course, to the IMBRSea for granting me the opportunity to partake in this really great master's program. And finally, thank you everybody for listening.